that are unable to get to here today. Welcome to another session of the Bioinformatics and Genomics series, from data analysis to AI applications. Today is going to be not me talking, but an invitee of the name of Simona Merlini, which is part of the Roberta Diaz-Brinton laboratory, or I would like to say the director of the Center for Innovation in Brain Science. Simona has extensive knowledge of R and using RNA sequencing data for research, and has lent us her attention for today's attention and knowledge for today's workshop. So without any further ado, I think I can hand this over to Simona, where you can share your screen perhaps. Or actually, before you do that, Simona, I did want to mention that for today, maybe I can put it in the chat. I'm going to copy this, and this is our home, where you can find all of the information on the workshop series, as well as the content that we do cover. And as you do remember from last week, we did cover the beginning guide to RNA-Seq, and Simona is here today to tell us even more about RNA sequencing. And for the material today, please do click on the material link recording over here, which will lead you to RNA sequencing data analysis in our workshop, which is what we're going to be covering today. And now, without further ado, I think you can take it away, Simona.
Thank you, Michele. Okay, you should be able to hear me right now. I'm going to share my screen. Okay, so if you follow the link that Michele sent in the chat, you will be able to go to the GitHub homepage of the workshop. And then from there are two main links I want you to open. There's the wiki link for the workshop material. In the meantime, I can send also this link in the chat. Oh, okay, perfect. Michele already done it. And then there's the art demo link that we are going to discuss. So if you open the wiki link, there's the home of the workshop. So the idea of this material is that you can find all the introductory information that you need to know about RNA-Seq and analysis in R, but also further materials if you are more curious to explore these themes. So today we're going to discuss a little bit about RNA-Seq methodology, and then we are going to focus on the differential expression analysis in R. We're going to explore two packages, the Lemma and the Seq2. And then we are going also to hopefully explore a practical guide and essential step with some tips for each of the steps in order to let you perform the all the pipeline to obtain differential expression analysis in R. So overall, no matter if your background, you should be able to follow along with this workshop. Again, based on your knowledge, you can also explore more materials. So overall, the main objectives is, again, to talk a little bit about RNA-Seq, and then summarize the key principle for differential expression analysis, that we're going to call it DGE, and then do a practical analysis in R with Lemma and DSC2. And finally, I want to give you some knowledge in order to understand your data, interpret your results, and visualize your results if you need to present your analysis or your data to someone. And to do this, we are going to open the introduction to RNA-Seq and differential expression wiki page. That is, oops. I'm going to move this bar over here. Okay. So briefly, what is RNA-Seq is a high throughput sequences technique that capture a picture of the gene expression of your samples. Today is a very used techniques because it's high sensitive, it gives you a lot of discovery power, and it allows you to do comparative analysis. Just to give you an idea, RNA-Seq technology involve a lot of steps before giving you the entire pictures of your samples. There is, first of all, a big step for sample preparation where you need to extract the RNA from your sample. And then there is all the sequences process, and then read alignment. After all these steps, there is the quantification of your samples. That means count all there is to estimate the gene expression levels. So for this workshop, we are going to start from the quantification of our samples in order to perform differential expression analysis. So there is two main way to do RNA-Seq. The first one is bulk RNA-Seq, where you capture an average gene expressions of the of all of your samples. And then there's single cell RNA-Seq that is able to capture the gene expression at the individual cell level. So the first question you should ask yourself when you have your data is, is my data coming from bulk or single cell RNA-Seq? For this demo, we are going to use single cell RNA-Seq data. And so before performing differential gene expression analysis, let's define what is this analysis. So DGE is a statistical approach that is used to identify genes whose expression levels vary significantly between conditions. So with DGE, you can reveal genes that, for example, are upregulated or downregulated in a condition of your samples. Usually, what you want to compare when you do DGE can be people that are diagnosed with a pathology or with a disease versus people that are not diagnosed with this disease. That will be the control. Sometimes you want to compare as a treatment. So you want to compare people that underwent under a specific treatment versus people that don't. And sometimes you want also to compare two treatments, for example. So every time you want to compare the gene expression of a condition versus a control, you can perform DGE. And so going on the second chapter of today, that is data preprocessing and normalization, what I do really want to stress before performing DEG is that especially for DEG, preprocessing and normalization are very important because since with DEG you are performing a lot of comparisons, you always obtain some results from this analysis. But the quality of your results and the reliability of your results depends on how you preprocess your data. So you do really need to know your data before performing DEG. And as I told you, we are starting with the quantitative results from RNA-Seq. That means that we are going to use a read count table where for each of the row you find a gene, and for each of the column you find one of your samples. So before performing DEG, you want to have your data looks like this read count table, and then you need to have a meta table data that gives you information about your samples. For example, information about the age of your sample, sex, or other variables you are interested to do your comparison. As I told you, if you want to compare the gene expression of a disease population versus the control, in your meta table, you need to have the disease information, of course. And so now, giving this background information, we can start looking at how to perform these in R. So if you open the demo, you can find the script. So first of all, this demo use data from this article in which the authors studied the asthma condition, and in particular, they explored how the use of glucocorticoids that is called, where is the name? Here, yes, dexamethasone, how the gene expression of people that were treated with these glucocorticoids changed compared to the control. And in particular, they found that the inflammation was mainly carried by this gene called CRISP-LD2. So again, we are going to see how to perform this analysis both in DS-SIG2 and Lima. So first of all, you can load all of the libraries. In the main pages of the GitHub, you also find the requirements file, in which you have also the script to download, to install all the required packages. And then we are going to import our read counts table, and you can access directly the CSV file from my repository. And then once you upload your data, you can see just visualizing the dimension of the dataset that you imported, that there are more than 38,000 rows and 9 columns. That means if you would open your dataset, you will see that, again, you have all the genes in the rows and all the samples in the columns. Of course, the dimension of this dataset is 9 for the columns, because the first column is the gene name. So for this demo, we have a total of 8 samples from human data and more than 38,000 genes. Now we need to import the metadata. And again, to do that, you can access directly the dataset from my repo, and then upload it and visualize the dimension and the dataset. So there's 8 rows and 4 columns. The rows in the metadata table are, again, the participants, and so our samples. And the columns are the variables that can be used to do the comparison. For these, we have DEX, that is the treatment. And as you can see, there are both control and treated. And in particular, there are 4 people that were treated and 4 people that were not treated. So the comparison is made comparing 4 samples, 4 gene expression samples of people with that were treated versus 4 controls. Then there is information about the cell type and the GOID, but we are not going to use this information for our analysis. So one of the very first things that you need to be aware when you upload the data is if there's some missingness in your data set. So missingness in RNA-Seq can be due to some technical issues, but also genes that were not able to be detected. So you need to understand the nature also of your missingness. So first of all, we are going to check if we have some missing values in our gene expression, so in the first table. And here we can see that we do not have any missing data because the count is 0. But usually you may found some missingness, and so there's missingness in DEG still hot issues, I would say, because there are some people that do not recommend imputing the data, others that do recommend. What I would say is that usually you can either impute your data or use the hypothesis that if a value is missing, you attribute to this value a zero value, that means no expression. But overall, even if you decide to impute your data or to change the missing values with zero, I would recommend to perform two parallel analyses, one using your imputation data and one using the data without imputation. And so to and the second one will get rid of all the missing values, so that at the very end you can compare your results and see also which is the effect of your imputations. Something that usually you can also do is get rid of the genes that have a missing more than 20% of the samples. So you can put again a threshold that is 0.2, that means 20%, and then get rid of the genes that has more than 20% of missingness. We do not have missingness again in this data set, but what you can also explore before analyzing your data is, since in this data set there is no missing data, I ask myself how many genes has expression that is equal to zero. And so I found out that there are more than 13,000 genes that has all the values equal to zero. That means that has all the 8 samples as a value equal to zero for these genes. So these genes are not carrying information for our analysis, so we can get rid of them. And so by removing these 30,000, more than 13,000 genes, at the very end of our analysis, we have now 25,258 genes for 8 samples. And again, here I look at the head of our data set. And then based on which package you use for performing DGE, you need to perform different transformation of your data. And before discussing about this transformation, we need to look more at our data set in order to know more about our data. So one thing that it's usually done is visualizing the data. And to do that, it is very useful to log transform the data in base 2. And here's the reason why, because if you plot, this is the box plot of the gene expression for all the 8 participants. So we have the participants on the x-axis, and then on the y-axis we have the mean gene expressions. And as you can see on the in the figure above, it's hard to understand the gene expressions. But then if you log transform your read counts, you can skew the data, you can skew more the data, and you are able to appreciate the gene expression more easily. So every time you want to do some kind of visualization of the data, the log2 transformation is very useful. And so depending on how you collect your data, if you bought your data from some company, or if you access data that you did not directly collected, you should be able to access the documentation where they do inform you about all the variability sources of your data. And so before, again, analyzing the data with DGE, we also need to see the variability of our data. So there are two main ways you can do it. One is plotting two gene expression samples. So we have one participant on the x-axis and one in the epsilon in the y-axis. And from this plot, you can see if there is some heteroscedasticity in our data. And in case you need to get rid of that. Another easy way to explore this is plotting the mean expression of the genes versus the standard deviation, and you can see if the variance depends on the mean or not. And then again, using the data that are logged to transform, you can do some pattern exploration of your data in order to understand any source or variability of the data, but also to detect some outliers maybe that we can see in the gene expressions. So the very first thing that you can do is do the pairwise correlation analysis, where you plot all the participants and you look if there's some correlation in the gene expression of the participants. You do want a high correlation, a high correlation score, because if the gene expression of the participant is very different, so not that much correlated, it may be due to technical issues in the collection of the data. And here you can focus on the first half of the plot. You see that overall there is a 0.97, 0.98 correlation, and that is good. You do not want a correlation of 1, of course, because it means that all the samples have the same gene expressions. And then you can perform hierarchical clustering just to have an idea of how your data can be clustered. Here we have few samples, and it looks like there's 2 participants that cluster together and do differ from the other 6. And then there is one of most used analysis that is the PCA, that is the principal component analysis, that is basically a dimensionality reduction that is able to summarize the variation across the samples using few components. So each of the components that you obtain from this analysis represents a portion of the total variance in the data. So after you can perform PCA on your log to transform gene expression, and then you can plot all the components of the first components of the PCA, and you can see that PCA1, that is the first bar of this plot, is carrying almost 40% of the variance. That is a good result. Whereas, for example, component 2 is carrying, I would say, less than 15% of the variance of the data set. A further analysis that we can do with the principal component is plotting the first 2 components. And so if we are plotting the data using principal component 1 and principal component 2, we can see how the 8 samples are distributed in this new space. And this is good because if we color the samples by the treatment, we can see that the pink one, that are the control, kind of cluster together and are far away from the treated one, that are more spread in the space. That means that maybe are carrying more diversity between among the same group. So overall, from this plot, we can understand that the conditions, so the treated versus control, are able to separate the data. So we are expecting to have different gene expression profile of the treated versus the one for the control. separated the data by, for example, stratify them for the treated or control condition, but also the cell type, for example. And again, the more metadata you have, the more variable you are interested in, you can stratify the plot with the information that you want. Okay, so now that we have cleaned our data, we have also explored a little bit about the variability and the variance of our data. We can perform DGE. So we are going to do it again using both Lemma and DSeq2. And one way to choose looking at how many samples you have. So Lemma is recommended for any data set, no matter the number of samples, whereas DSeq2 is recommended and works very well for data set that has less than 12 samples. And then there is another package that is not in this demo, but you can find more information in the wiki material that is called EdgeR, and is better for larger data sets and with more than 12 samples. So since we have only 8 samples in our data set, we're going to look at DSeq2 and of course Lemma. So each of the packages has its own way to pre-process the data. So for Lemma, if you want to use Lemma, you need to know that you need to log transform the data before performing the analysis. And here is there are some lines of code in which you can create you can log transform your data. And here we can see that the new data set with the values that are log transformed for each of the genes. And then once you have log transformed the data ready for being analyzed with the Lemma. To do that, what you need to give as an input of the function that is called lmfeed is giving your object that is the log transform data set, and then create a design of your model. That is the only way you can control the variability of your results. Because with DGE, so with differential gene expression, what you do is compare for each of the gene expression, compare the gene expression of the treated versus the gene expression of the control, and then ask if there are some differences between these two gene expressions profile, and if this difference is significant. So you are basically doing a linear regression analysis, and with the output variable that is the gene that you are, that is one of the 25,000 genes that we have in our data set. And then with the input variable that is the condition. So if you know that for the setup of your experiment, you have other sources of variability. Sometimes these variabilities can be the age of the participants, but also sex. Other times can be more technical, so it can be the platelet dimension that they use for the RNA-Seq. So based on the variability you don't want to include in your results, you can add more variable on your design. So here, since we only have few metadata and we do not have data about the RNA-Seq process, we are doing only a univariate regression model. So for each of the 25,000 genes, we are doing a linear regression, regressing out the information of our group. That is, again, treated or control participant. And then you can obtain the output of the lemma that gives you for each of the genes that again you can see in the rows of the table. You have the log-fold change, the average expression, and other information that I'm going to discuss. So one of the most important information that you have from this analysis is log-fold change. That is the log2 transformed fold change in expressions between the two group of comparison that are treated versus control. So a positive value of the log-fold change means that that gene is overexpressed for our example in the treated group compared to the control. And if you have negative values of the log2 fold change, it means that that gene is underexpressed in the treated group versus the control. Another things that you want to consider is the p-value, because as I told you, you are doing a linear regression, and then what you want to see is that if that log-fold change that you obtain is actually significantly is a significant score. And so you want to look at the p-value or the adjusted p-value. So usually if you have, as in our case, that we have more than 25,000 genes, and we are doing a comparison for each of the genes, you want to use the adjusted p-value that is able to take into consideration the fact that you are doing multiple comparison. If you have very few genes, you can also use the p-value. And then there's other information like the T and the B that are the measure the score that are used to the statistic to the test that you use to see if the differences in the treated and control is statistically significant. So once you have your log-fold change for each of the genes and then the adjusted p-value, you are kind of done with the differential expression analysis, because you can filter this list by considering only the significantly different genes. And then you can, here's a way to filter these genes. One thing again that you can ask yourself is which threshold should I use to filter these genes? And again, it really depends on the number of comparison that you're doing. And if there's, as in our case, more than 25,000, we are considering the adjusted p-value. And then you can either use 0.05 as a threshold or 0.01. One thing that you can do is to try to filter with both thresholds, like 0.01 and 0.05, and look at the numbers of differential expressed gene that you obtain, and if these numbers really change between the threshold that you put. And then we are going to do the same things with DSeq2, but keeping in mind that DSeq2 does not need log2 transform data as an input, but before performing the DEG, you need to transform the data based on the size factor normalization. So here's the code that you need to apply in order to do the size factor based normalization. And once you have normalized the data with the size factor, you can perform the differential expression analysis with DSeq2 that then is going to automatically in this function that is called DSeq is going to apply the normalization in order to obtain the differential expressed gene. So in the same way, you are going to obtain the same information that we discussed earlier, so the log4 change and the p-value and the adjusted p-value, but something more interesting that DSeq2 gives you as an output is a summary of your analysis. So this is an example of the output that we obtain. And it tells you that out of all the 25,000 and more genes with non-zero total read count, there is taking as a threshold the adjusted p-value that is less than 0.1. There is it gives you the count of upregulated genes, and then the count of downregulated genes. And then it's also able to detect the outliers, and it gives you the count of probably some of the outliers that you need to take into consideration, and then other genes that are not significant. And so now that we have both the results from the lemma and both the results from the DSeq2 packages, we can look at how to examine and visualize the results. So for this visualization part, we are going to use both results. So one thing that you can do is look at how differentially expressed gene you have obtained with lemma, and how many you have obtained with DSeq2. To do that, you can perform a Venn plot. And here, for example, we have obtained 1,183 genes that are shared both in the DSeq2 results and in the lemma. Only 19 were obtained only in lemma, and almost 1,000 were obtained with DSeq2. So there were way more differentially expressed genes obtained with DSeq2 compared to lemma. And you can visualize the same information using the upset plot, where you can where you have the first bar that is the number of differentially expressed genes shared among lemma and DSeq2, and then the one only with DSeq2 and only with lemma. And so for this visualization part, we are going to use only the differentially expressed genes obtained with DSeq2. So the I would say most used plot to visualize the genes that we obtain is called the volcano plot. Here's the script that you can use to visualize it. And I just want to spend some words to explain you how to interpret this plot. So first of all, on the x-axis, you have the log2 fold change. And in the y-axis, you have the minus log 10 p-value. So with the y-axis, you can choose the threshold of your p-value. And here, the threshold is 0.05 with the adjusted p-value. So you can see horizontally that the data are divided. And then you can see that all the red plots that are going on the right part, all the red dots that are in the right part of the plot are all the genes that are upregulated and are significantly upregulated, taking into consideration the 0.05 adjusted p-value. And also here, you can also choose the threshold of the up and down regulation. So you want, for example, to show only the genes that are at least double over or under expressed in the treated group. And the same in the blue dots are all the genes that are significantly downregulated in the treated group compared to the control group. And so this plot gives you an idea of where to put the threshold of your p-value, and to see if changing the threshold of your p-value include or exclude how many genes. And then you can also see the magnitude of the log two-fold change. At the very right part of the plot, there's the most over upregulated genes, and the same for the left part with the downregulated genes. Another plot that is less used but can give you an idea also of possible outliers of your analysis is the MA plot. The main changes with this plot is that you don't have anymore the log two-fold change on the x-axis, but now you have it on the y-axis. And then on the x-axis, you have the average expression of your samples. So you can see the down and up regulated genes and how their expression is in your data set. And I said that with this, you can see also outliers, because if you have dots that are at the very right of your plot, they may be outliers, especially if they're very far away from the previous dots. And then you can also, and this is a very useful way to summarize your results. You can perform heat map of the differentially expressed genes. And so you can, again, I'm not going through all the lines of the script. You can find it and run it on your own code or with this demo too. So with the heat map, in this case, we have all the 8 samples as the column of the heat map, and then the rows are all the genes. Here I plotted only, I would say like the first 40, no, not 40, I would say 20. Yeah, the first 20 most expressed, most dysregulated genes. And be aware that there's a scale, a color scale. The red one is overexpressed in treated versus control, and the blue one is underexpressed in treated versus control. And you can see all the shadows that helps you understand the dysregulation in your data set. And you can also add some metadata, for example, the treated and the control variable. So the green one are the control, and overall, you can see that in the control, there is downregulation of the genes that are overexpressed in the treated control. So that means that in the treated, there's all the dysregulation, most of the dysregulation is driven by the upregulated genes. And this is easy to do if you have 8 samples, because you have only 8 columns. But what if you have more than 8 samples? So if you have more than 8 samples, it can be difficult to create a column per sample. So you can just plot the average expression of, in this case, the treated group and the control group, being aware that the scale of your log fold change can change, because here there's with this plot you are more able to capture the magnitude of the log fold change, because you are plotting each of the results for each of the participants, whereas here you are using only the mean information, and our range for the log fold change is different and is a little bit smaller. But again, here you can for sure appreciate and also more easily appreciate the fact that the treated group is characterized by an overall upregulation of the dysregulated genes. And then this is everything I wanted to discuss with you as far as understanding the setup of the analysis, understanding the data, and then analyzing them And once you have the output, how to visualize all the results. The very last things I want to discuss with you, since I have a little bit of time, is the name of the genes. So in this analysis, as we can also see in this plot, the gene's name is coded with this code is ENG, SG, and then many numbers. So another thing that you need to take in consideration is how your gene are coded in your data set. And here I implemented a small function that can tell you if you're not if you're not that familiar with gene names. There is a function that tells you which type of gene name you have in your data set. And so this function is checking if your names of the genes are coded with symbol or entrance ID, ensemble, or RefSeq, that are the most used code for genes. And in our case, the function told us that our gene names are coded with the ensemble code that is this code that you can see also in the heat map. So what if we want to have a more readable results? We can convert all the gene that are coded with the symbol with their actual name. So there is this function that is able to match for all of the ensemble code the gene names with this that is called symbol. And so you can match all the genes name. And here you can see, especially for if you have a strong biological background, the gene symbols should sound more familiar to you, because you can actually see the gene names. And you can show the results with these symbols. And for this specific example, what I did is to plot again the volcano plot, but with a subset of gene names I was interested in, that were specifically the one that were found by the authors of the article. And I don't know if you remember, but there was the gene called CRISP-LD2 that was found as one of the main drivers of the inflammation in the treated group, that I don't know if you can read it, but we found it as one of the significant overexpressed, so upregulated gene in the treated control. So this is also a double check that you can do if you are interested in a particular subset of genes, and that's an easier way also to visualize the dysregulation of your gene of interest. Okay, so with this, I'm done with the demo. And again, I want to remember you that if you want more information, you can either ask me or look at the materials that in which you can see a more breakdown of all the demo that I've done with more information. But thank you for listening to me, and I'm open to question if there is any.